Good afternoon, friends. I'm Solomon. Today I'm with new video which contains part 2 of chapter 3, and this part mainly focus on the plasma membrane or cell membrane and transport across cell membrane. Mainly active transport and passive transport will be discussed in detail. So as usual, don't forget to like, share, comment, and subscribe this channel. Thank you very much, and let us continue to the presentation. Well, this is part 2 of the chapter 3 because chapter 3 is long chapter, so that's why I'm classified it into two groups or two parts. Therefore, in this part, we're going to see plasma membrane as well as the mechanism of movement of molecules in and out of the cell. Therefore, these two subtitles or ideas will be discussed under this part of the chapter 3. Anyways, this is a plasma membrane or cell membrane. Cell membrane is the outermost boundary of living cells. Even if some cells, for example, plant cells, fungal or algal cells have additional or extra boundary, which is cellular. However, cellular is not the living part of the cell, but the living part of the cell starts from the cell membrane. Therefore, we say that the cell membrane or plasma membrane is the outermost boundary of living cells. Second, this plasma membrane or cell membrane encloses the intracellular fluid of the cell. The intracellular fluid, the fluid which is found in the cell, is enclosed by the cell membrane or plasma membrane. This plasma membrane or cell membrane is selective permeable membrane. It selects what enters and what leaves in and out of the cell. Things are selected to be taken into the cell or to be released out of the cell by the plasma membrane. So it is selectively permeable membrane. All plasma membranes are made up of three molecules, especially the macromolecule. The lipid, protein, and some amount or small amount of carbohydrates are also used to make plasma membranes. Specifically, the lipids are phospholipid types. As you know, phospholipid is made up of three main components: one glycerol, two fatty acids, and one phosphate group. Therefore, for common understanding, the structure of phospholipid looks like this picture. For common understanding, for example, the head part is phosphate part, which is polar in nature because it is negatively charged. Yeah? So it is polar. The two tails are fatty acids, which are non-polar molecules. Electrically, they are neutral in nature. Therefore, the lipid types for plasma membrane formation are phospholipid, as well as this amount of cholesterol also there for the formation of plasma membrane. So lipid, specifically phospholipid and cholesterol, protein, and a small amount of carbohydrates are used to make all plasma membranes. As already said, the phospholipid has two main parts: the polar end part, which is hydrophilic or water-loving part, and the non-polar end or the hydrophobic part, or it is water-fearing or water-hating part. So whenever this molecule, the phospholipid molecule, is mixed with water, it doesn't mix with water. Therefore, whenever these phospholipid molecules are come together, they assemble themselves in the lipid bilayer form. That means they form double layer of lipid molecules when they contacted with water. Whenever they contacted with water, they make or they arrange themselves in lipid bilayer form. The hydrophilic part or the hydrophobic part, the fatty acid part, they are bury themselves in the center, away from water. Both sides, the hydrophobic parts are arranged themselves to the center, away from the water. But the head, the polar end, the phosphate group part, is facing itself towards water. Here, this is water. This one is also water. Therefore, the polar end facing themselves towards water. So this arrangement makes the cell membrane to have or to has a lipid bilayer arrangement. Therefore, the structure of a cell membrane is mainly determined by the nature of phospholipid of its parts. Therefore, the cell membrane is structurally looks like lipid bilayer forms. Phospholipid is a fluid which looks like a liquid cooking oil. Therefore, this cholesterol lays in between phosphate molecules. For example, in this figure, this is cholesterol, is another cholesterol, another cholesterol, another cholesterol. So cholesterol molecules are situated or laid in between the phosphate group of phospholipid molecules. So being there, they have provide a number of functions. For example, cholesterol can prevent the fatty acid chains from packaging together. So the fatty acid chains are packed together whenever cholesterol is found there. And also this cholesterol crystallizing the membrane to decrease its fluidity. The fluidity of the membrane is determined by cholesterol. When the amount of cholesterol increases, the fluidity of the membrane decreases. When the content or the amount of cholesterol in membrane increases, so the fluidity of the membrane decreases. Therefore, it determines the fluidity of the membrane. Fluidity of the membrane gives flexibility to the cell to change their shapes. And also transport processes are determined on the fluidity of the lipid bilayer. Therefore, indirectly, transport process through cell membrane as well as flexibility of the cell to change their shapes are indirectly determined by the amount of cholesterol found in cell membrane. Here, another molecules are found in cell membrane. For example, a number of proteins are found here. Peripheral protein is there. Integral protein is also there. Cytoskeleton protein networks, which provide the shape and the size of the cell. And also, these are channel proteins through which ions are passed. Cholesterol is here too. And carbohydrates, specifically glycolipid molecules, as well as glycoprotein molecules, also found throughout cell membrane. All these molecules have their own specific functions. In the next slide, we'll try to discuss the functions of these molecules. To summarize, cell membrane composed of the following molecules. For example, the first molecule is lipid bilayer. This lipid bilayer includes phospholipid as well as cholesterol. So this lipid bilayer forms the basic structure of membrane. It is a barrier to pass of water-soluble substance between the intracellular fluid and the extracellular fluid. It is responsible for the fluidity of membrane because fluidity of the membrane is determined by lipids, specifically by cholesterol. The other content or the other component of the cell is membrane proteins. There are a number of membrane proteins are found in a cell membrane and have their own specific functions. For example, channel membrane protein. By the way, MP means membrane protein. They form passageways or channel across the lipid bilayer and allow specifically ions to pass through without direct contact with the lipid interior part. They make a channel, a tube, through which ions pass without directly contact with the rest part of the lipid membrane or lipid part of the membrane. Therefore, this channel or this tube is channel membrane protein through which ions are passed. Second, these carrier membrane proteins. These carrier membrane proteins are proteins serve as carrier molecules. They carry molecules and transport specific molecules, specifically solids that cannot cross on their own. Because of different reasons, these ions cannot pass through by their own through cell membrane. Therefore, they need carrier molecules. So these carrier membrane proteins are very important to carry and transport this solid through the cell membrane. The other is receptor membrane protein. These proteins are found on the outer surface of membrane. They recognize and bind to specific molecules such as hormones, neurotransmitters, growth factors, cell adhesions, and different nutrients. So by recognizing such molecules and they receive them and help this molecule to pass through cell membrane. The other molecule is membrane-bounded enzymes. There are enzymes found in the either side of cell membrane. This is to catalyze any chemical reactions that carried out on the either side of the membrane. Therefore, these are enzyme types. They are protein types. As you know, all enzymes are proteins. Therefore, they catalyze different chemical reactions on the either side of the membrane. Plasma membrane. Filamentous network membrane proteins are another protein types which are found in the inner side of the membrane, and they are secured to the internal protein elements of the cytoskeleton. The cytoskeleton is, or cytoskeletons are secured by these filamentous networks of membrane proteins. The other protein types are adhesion proteins. These adhesion proteins are protein types functions as cell adhesion that protrude from the membrane surface that grip each other and grip to the connective tissue. That means the cells are gripped to each other, or they are joined together, and as well as they are grip or connected with the connective tissues. Therefore, this adhesion action is carried out by adhesion protein molecules. The other molecule or component of cell membrane is glycoprotein. Glycoprotein is formed from glycogen and carbohydrate and protein molecules. Some proteins conjugation with carbohydrate to form glycoprotein. They are important in the cell's ability to recognize self and in cell-to-cell interaction, because in level of organization, different cells come together to make tissue. Then related tissue come together to make organ, and the related organ come together to make system, and the systems come together to make organism. Therefore, similar cells that have the same functions will come together to make respective tissue. Therefore, to recognize to each other, to know one another, and even in cell-to-cell interaction, these molecules, glycoprotein molecules, are very important molecules. They are also known as membrane carbohydrate. We can say they are membrane carbohydrate or glycoprotein molecules. Here we can see different molecules are found in cell membrane as a component. For example, this is the lipoprotein, bilayer molecule of cell membrane. And intervally, there are different molecules are situated. For example, this is receptor molecules. These receptor molecules receive, by recognizing the different molecules, they receive. These are enzymes. These enzymes are catalyzing chemical reactions on either side of the membrane. This is channel through which ions pass. These are cell identifying markers, specifically these glycoproteins. Glycoproteins are important for recognizing or to identifying cell one another. Still, these are cell adhesion molecules that connect, that grip the cell one another, or the cell to be gripped with the connective tissue. So these molecules are very important to adhere the cell in its place. Well, in this subtitle, we'll see the cellular diversity. Cells are very diverse in their size, in their shape, as well as in their internal structure. For example, based on their internal structure, cells can be classified as prokaryotic cells as well as eukaryotic cells, because the prokaryotic cells have no true nucleus, as well as they have no membrane-bounded organelles. However, eukaryotic cells have true nucleus as well as organelles that are bounded with membrane. Based on cell shapes, cells can be classified they can be diverse based on their shape. For example, cells have different shapes. This is for their appropriate functions. For example, most body cells are flat. Most our body cells are flat. This is because they protect and cover the body surface. Nerve cells are long extensions because they are extended from our foot to the head, so they are long extension. This is for their specific function. Skin cells are flat in their shape, where egg cells are spherical in shape. Some bacteria are rod is their shape. Plant cells are rectangular. Some plant cells are rectangular in their shape. When it comes to the cell size, some cells can be seen without microscope. Simply, they can be seen with the naked eye without any microscope. For example, egg cells. Eggs of birds and eggs of reptiles, as well as neurocells of the giraffes. This can be seen without the microscope, but most cells are microscopic. They cannot be seen without microscope because they are very small in their size. This is transport across the cell membrane. Already we say that cells are basic functional units of organisms because important molecules that determine life of organisms are produced in the cell. Therefore, to produce such molecules that determine life, their raw materials must be entered or taken into the cell. Then after chemical reaction is carried out, the product must be taken out of the cell. Therefore, to take materials into the cell, as well as to bring out molecules out of the cell, transport mechanisms must be discussed. So here, different transport mechanisms are going to be discussed in detail. Transport mechanism. The permeability of substance across cell membrane is determined by their solubility in lipids, and a note on their molecular size. Some molecules can be soluble in lipids, but still some others cannot soluble in the lipid. So water-soluble compounds are generally impermeable, and they require carrier-mediated transport. Lipid-soluble molecules can pass through cell membrane. However, water-soluble molecules cannot be passed through cell membrane. Rather, they need carrier-mediated transport. An important function of cell membrane is withhold unwanted molecule while permitting entry of molecules necessary for cellular metabolism. This cell membrane can withhold unwanted molecule not to enter to the cell, but it permits any molecules that are necessary for cellular metabolism. Transport mechanisms can be classified as passive transport, which includes simple diffusion, facilitative diffusion, ion exchange channel, and osmosis, and active transport, which includes pumps. Simple diffusion is a type of passive transport. So in simple diffusion, some solids and a gas enter into the cell simply passively. They are driven by the force called concentration gradient, because there is a concentration difference between the internal and the external part of the cell of that specific solids or gas. Therefore, because of this concentration gradient difference, so these molecules can be driven into the cell. The rate of entry is proportional to the solubility of the solutes in the hydrophobic core of the membrane. The hydrophobic core of the membrane is lipoprotein, the lipid part or the phospholipid part of the membrane. Therefore, all solids have no equal potential of solubility in lipid molecules. Therefore, high soluble solids can diffuse faster than less soluble solids in lipid molecules of the cell membrane. The net movement of molecules by simple diffusion will be from high area of concentration to lower area of the concentration. For example, here in this figure, before a diffusion carried out, the concentration of molecules is high at the extracellular part of the cell. But when time passes, these molecules are diffused small in the small. They diffused into the cell small in the small, small in the small. Then after, at the end of diffusion, the net movement of these molecules is almost zero, because at equilibrium there is no net movement. Therefore, the simple diffusion will be from areas of high concentration to areas of low concentration until equilibrium constant is or until equilibrium is attained. The molecules move down their concentration gradient, meaning the net movement of molecules is from areas of high concentration to areas of low concentration. Because every molecule has their own or its own kinetic energy. That's why they are moved here and there, but the net movement is considered in diffusion. In simple diffusion, there is no requirement of energy, so cells, they do not expand any cellular energy for simple diffusion purpose, and it is relatively slower process. Okay, this is movement along electrical gradient. Diffusion of, simple diffusion of some molecules may be affected not only by the concentration gradient, but also by their electrical gradient. So both the concentration gradient as well as the electrical gradient may affect the diffusion of some molecules. The movement of charged particles is also affected by their electrical gradient. These charged particles, the movement of these charged particles can be affected both by their concentration gradient as well as by their electrical gradient. If a relative difference in charge exists between two adjacent areas, therefore the cations, these cations are positively charged particles, whereas anions, negatively charged particles. Therefore, this cation, which are positively charged particles, tend to move towards negatively charged area. The net movement is towards the negatively charged area, whereas the anion, which are negatively charged particles, tend to move towards positively charged particles or positively charged area. If both the concentration gradient and the electrical gradient exist for a particular ion, these together are known as electrochemical gradient. Well, facilitative diffusion is another type of passive transport. This is carrier mediated transport. This facilitative diffusion is carrier mediated transport. We know that membrane transport involves specific transport proteins such as channel membrane proteins as well as carrier membrane proteins that are embedded in the phospholipid bilayer. So solids that are not soluble in lipids can pass through carrier proteins of the membrane. These channel proteins are important to channel or to transport ions. They transport ions, but the carrier proteins are important to carry and transport solids that cannot be dissolved in lipids. The passage of solids through carrier membrane, carrier proteins in a cell membrane is known as facilitative diffusion. For example, glucose transport across membrane is facilitative diffusion, which involves a family of proteins known as glucose transporters. These are glucose transporters are protein types or carrier membrane protein types that transport or that carry glucose to pass through cell membrane. Ion channel is another passive transport type. Membranes have specific device or proteins called ion channels that are important for channeling or transporting different ions. Ion channels are transmembrane proteins. They are what? They are transmembrane proteins that allow the selective entry of various ions. These channels are for quick transport of electrolytes or ions such as calcium ion, potassium ion, sodium ion, chlorine, and the likes. These are selective ion conductive ports. The channels generally remain closed, but in the response of stimuli, they open, allowing rapid flow of ions down their gradients. Whenever these ions are not needed, these channels are closed. But when they receive some information, or when they are triggered by some stimuli, immediately they are open and allowing for the flow of ions down their gradient. Therefore, in this way, ions are transported or move into the cell through these ion channels. Well, osmosis is another passive transport, and it is a special type of diffusion. Specifically, it is the net movement of water molecules down its concentration gradient. It is a special type of diffusion for diffusion of water molecules. As any diffusion, this osmosis is the movement of water molecules down its concentration gradient. For instance, we can see here three types of cells. One of the cells is or has the same concentration content or the same concentration nature with its external fluid, the intracellular fluid of the cell, and the extracellular fluid of the cell have the same concentration, so there is no net movement of water molecule. So we can't say that in this condition water is diffused. So there is no osmosis because the intracellular fluid of the cell and the extracellular fluid of the cell have the same concentration. In other cell types, if the intracellular of the cell has more concentrated than the extracellular fluid, if the extracellular fluid is less concentrated than the intracellular fluid of the cell, immediately water will be diffused from outside to inside. It means from the extracellular fluid to the intracellular fluid. This type of concentration is known as hypertonic solution. I mean, the intracellular fluid of the cell is in hypertonic solution. So water will be diffused from outside, from extracellular fluid to intracellular fluid. In other words, if the intracellular fluid of the cell is less concentrated, or if it is in hypertonic solution conditions, so the water molecule are diffused from the intracellular fluid to the extracellular fluid, from inside to outside. Therefore, these are three main conditions in which water molecules are diffused accordingly. The driving force for diffusion of water, or for osmosis, is the concentration gradient of the water between two adjacent areas. Always the water molecules are diffused from their high water concentration or low salt amount to the areas of low water concentration, or into the hypertonic solution. Hypertonic solution. So osmosis doesn't require any expenditure of energy. Water can be, or we can say that high water concentration gradient, water concentration molecules, or you can say high water potential, low water concentration molecules, or low water potential. So these phrases can be used interchangeably. Active transport is another transport mechanisms of molecules in and out of the cell. So this active transport is a type of transport which requires energy expenditure. But in passive transport, there is no need of energy for transportation. But here in active transport, energy is required. Second, this active transport, 14% of the expenditure of energy in the cell is used for the active transport purpose. Only 16% are used for other purpose, but 14% of energy lost or energy expenditure is for the purpose of this active transport. The active transport is unidirectional. The transport or the movement of molecules or materials are in one direction. But in passive transport, molecules may be transported here and there in both directions, but the net movement is considered. Therefore, for active transport, molecules are substances are moved unidirectionally. The transport of molecules is from areas of low concentration to areas of high concentration. In passive transport, molecules are the net movement of molecules is from areas of high concentration to areas of low concentration. That means toward this concentration gradient or along concentration gradient. But in active transport, it is from areas of low concentration to areas of high concentration or against concentration gradient. In active transport, molecules or substances are transported from low concentrated to high concentrated. From low concentrated to high concentrated areas. Always unidirectional, and they are moved from low concentration to high concentration. This type of transport is especially it needs or it requires special integral proteins known as transporters. Without these proteins or transporters, molecules in active transport cannot be transported. So it needs what? Transporter proteins, which are integral proteins. The transport system is saturated. At some area, these transport proteins may be saturated. So the next molecule to be transported have to stay and wait for it to start. Once these transporter proteins are saturated, so the next molecule to be transported have to stay and wait for it to start. The transporters are susceptible to inhibition because there are molecules which are resemble with the molecules that are transported. So these molecules may occupy these transport proteins. So if these transport proteins are occupied by inhibitors, then after the molecules, the real molecules that used to be transported have no chance to be transported. So there may be inhibition of these transport proteins. Therefore, the active transport may be affected by the inhibitor molecules. These are some nature and characteristics of active transport. Sodium-potassium pump is a good example of active transport. The major ions within the cells and their surroundings are sodium ion, potassium ion, as well as chlorine ion. These ions are found in the cell as well as their surrounding cell surface. The studies have shown that cell surface membranes of most cells have sodium pump, which is coupled with potassium pump. So the combination of these two pumps is known as sodium-potassium pump. The pump is a carrier protein. It is a protein type which is specifically carrier protein, which is found or situated in cell membrane from one side to another side of the membrane. The transfer of sodium and the potassium ions across cell membrane brings a change in the shape of these carrier proteins. This sodium-potassium pump plays three important roles. For example, it establishes sodium and potassium concentration gradient across the cell membrane of all cells. These concentration gradients are important in the nerve and muscles to generate electrical signals, electrical messages. So this is the first function or importance of this pump. Second, it helps in regulating cell volume, because the cell volume is determined by the solution it contains. The solution is formed from solutes and water. Therefore, solutes are if the solution concentration of the intracellular fluid is more than the extracellular fluid, so immediately water will be diffused into the cell, and the volume of the cell will increase, and it will swelling. If it is a plant cell, animal cell, and if more water is diffused into the cell, then the cell will be burst. But if it is a plant cell, it can resist this pressure because plant cells have a cell wall. But if the extracellular fluid of the cell is more concentrated than the intracellular fluid, so water will be driven out of the cell into the extracellular fluid. So the volume of the cell will be reduced, and the cell becomes shrink. So this way, this sodium-potassium pump can regulate the volume of the cells. Third, the energy used to run this pump, because this is active transport, so the active transport already requires energy. Therefore, this energy that is required to drive or run this pump is indirectly served as a source of energy for co-transport of glucose and amino acids across cell membrane. Specifically, cells of intestine and kidney can use this energy as indirect source of energy to drive or to transport glucose and amino acids through their cell membrane. How this pump works? It brings sodium ion out of the cell and potassium ions into the cells. For example, if this is cell membrane or it is plasma membrane, this pump or it's a protein which is integrally found or situated in this plasma membrane. So in the first, this plasma membrane or this protein is found in the cell membrane, for example, and it looks like open at the inner part and it is closed at the outer part. So when immediately these sodium ions are, 3 sodium ions are bind to this protein or channel protein, and when the phosphate group is attached to this protein, because this phosphate group is found from ATP. That's why active transport requires energy in the form of ATP. Therefore, ATP is dissociated into ADP and phosphate group. Therefore, this phosphate group is attached to the this protein or this channel protein. So immediately its shape is changed, meaning its shape is changed into this way. It will be closed at the inner side and it will be open at the outer side. Still, this phosphate group is attached. Because of that, the shape of this protein is changed. At the inner side, the protein is closed, but in the outer side, it is open, so easily these 3 sodium ions are released to the external part of the cell. Simultaneously, when these sodium ions are released, immediately the potassium ions, 2 potassium ions are attached to the this protein. For example, if this color is color of potassium, 2 potassium ions are attached to this protein molecule. Whenever they are attached to this protein or channel protein, so phosphate will be released, meaning the released phosphate is joined with ADP and ATP, that is energy, is produced. So immediately when these phosphate ions are attached, and these potassium ions are attached, and the phosphate group is released from this protein, so its shape is restored back. So it looks like this. At the outer side, it will be closed, and at the inner side, it will be open. Therefore, these potassium ions are released into the cell. So in this way, the cycle will repeat itself. First, 3 sodium ions are attached and the phosphate group is attached. Immediately the shape is changed because at the outer side it will be open and at the inner side it will be closed. Then it releases 3 sodium ions. Simultaneously, these potassium ions are attached. When they are attached to this protein, so phosphate group will release from the protein. So its shape will be changed back to the original. So in this way, it brings potassium ions into the cell, 2 potassium ions into the cell, and 3 sodium ions out of the cell. So in this way, the cycle will continue, and it works in this manner. Vesicular transport is an example of active transport, and it is a special cell membrane transport system which transports ions, small polar molecules, even large polar molecules, as well as multimolecular materials such as hormones, even evading microorganisms or microbes, including bacteria. So such materials or such molecules can be transported in and out of the cell by vesicular transport. This vesicular transport is 2 types, exocytosis as well as endocytosis. Transport into the cell is endocytosis, and the transport out of the cell is known as exocytosis. Endocytosis is mechanism by which cells internalize extracellular macromolecules to form endocytic vesicles. For example, if this is extracellular macromolecules, to bring this molecule into the cell, immediately the cell forms endocytic vesicle. This macromolecule is enclosed by the cell membrane itself, by pinching off the cell membrane part, and it makes or it encloses the macromolecule that is going to be internalized into the cell. So this requires, or this type of transport requires energy in the form of ATP, because it is active transport. As already known, this active transport requires energy. So vesicular transport, especially this endocytosis, requires energy in the form of ATP. Endocytosis are 3 in type. Pinocytosis, which is cell ring, receptor mediated endocytosis, as well as phagocytosis, which is cell eating. In pinocytosis, materials are taken in the liquid form. In pinocytosis, formed vesicles are extremely small. They are micro in their size. So the process is known as micropinocytosis. The process of the formation of these vesicles in pinocytosis is micropinocytosis, because the formed vesicles are extremely small in their size, and the formed vesicles are known as micropinocytic vesicles. They are micropinocytic vesicles. Here, for example, these molecules are liquid molecules that are going to be taken into the cell by pinocytosis. So whenever they are taken into, before they are taken into the cell, they form vesicles. This vesicles is formed by pinching of the cell membrane itself, and by enclosing the liquid molecules into this part of the cell membrane, and make extremely small size vesicles. Then after once the vesicles are formed, they are taken into the cell. Receptor-mediated endocytosis is the second type of endocytosis. In this mechanism, materials or molecules are entered into the cell whenever they are attached to the receptor proteins on the surface of the cell membrane or on the surface of the cell. All liquids cannot be entered into the cell simply by pinocytosis alone. Rather, some liquids may need receptor molecules or receptor proteins that help them to attach to the cell and then after enter into the cell. So the process is known as absorptive pinocytosis. Several hormones are also taken up by the cell by a receptor-mediated mechanism. Many viruses get attached to their specific receptors on the surface on the cell membrane. You may heard HIV virus may not affect some individuals. Such individuals have no cell receptors, receptor proteins on the surface of CD4 cell or T helper cell, because HIV can attach itself to the receptor protein of the T helper cell or CD4 cells and enter into the cell and reproduce there. But individuals with which have not affected by HIV/AIDS have no such protein. So their CD4 cell or T helper cell is without such a receptor protein. Anyways, this virus, for example, influenza virus, hepatitis B virus, polio virus, and HIV virus too, they can enter into the cell through receptor-mediated endocytosis mechanism. Once they are attached to the receptor molecule and they make vesicle, they are attached to the receptor protein, and immediately the cell membrane is pinched off and make a vesicle. Then after, the vesicle will be taken into the cell. So in this mechanism, receptor-mediated endocytosis helps the cell to bring molecules into it. Phagocytosis is the third type of endocytosis. In phagocytosis, materials are taken in the solid form. Materials that are taken into the cell is in the solid form. Cells that carry out the phagocytosis process are known as phagocytes or phagocytic cells. You can use phagocytes or you can use phagocytic cells. And the sac, which is formed during the uptake of this solid form of molecules by a process known as phagocytosis, is called phagocytic vesicle. This phagocytosis is an important mechanism for phagocytic white blood cells to ingest these invading microorganisms. For example, this is solid material that can be engulfed or that can be ingested into the cell, but before it engulfed into the cell, it forms a phagocytic vesicle. This phagocytic vesicle is formed by pinch of the cell membrane. Then after, the whole material will be taken into the cell. Well, this exocytosis is another type of vascular transport, and it's the transport of materials out of the cell. So it is the reverse of endocytosis. In endocytosis, materials are transported into the cell, whereas in exocytosis, materials are transported out of the cell. For instance, waste materials, such as solid and undigested remains of phagocytic vacuoles or phagocytic vesicles, may be removed from the cell. Useful materials such as secretory enzymes from pancreas, for instance, may be achieved by the exocytosis. Meaning phagocytic cells, pancreatic cells can produce pancreatic enzymes. So the function of these pancreatic enzymes is in small intestine. Therefore, they must be transported from where they are produced into where they are functioning. They are producing pancreatic cells, but their function is in small intestine. Therefore, they must be transported by a process known as exocytosis. Not only enzymes, but also other materials, other important materials, for example, hormones, also produced, transported from where they are produced to where they are functioning. Even plant cells use exocytosis to export or to transport materials which are needed for the formation of cellule. This cellule is formed from cellulose. Therefore, cellulose which is produced in the cell must be transported to where cellulose, new cellule is formed. Therefore, this exocytosis is very important process to transport materials, either waste or useful materials, out of the cell to remove or to transport to their target or functioning areas. To summarize previously discussed transport mechanisms in this single table, here transport types, either the transport is active or passive, which molecules are transported by specific transport mechanisms, which direction does a molecule transported, does the transport needs energy or not, as well as is there a requirement of transport proteins for specific transport mechanisms. First, simple diffusion. It is a passive type of transport. Smaller and hydrophobic molecules can be transported through simple diffusion. The direction is down concentration to gradient or towards low concentration. Simple diffusion doesn't require both energy and protein molecules. Facilitative, osmosis is a passive type of diffusion. Specifically, water molecules are transported through osmosis. It is towards high concentration of solutions from less concentrated solutions. This osmosis doesn't require any energy and transport proteins. Facilitative diffusion is passive type of transport. Any molecules that require transport and can be transported. The movement is down concentration gradient, which is towards low concentration. Facilitative diffusion doesn't require energy, however, it requires transport proteins. Pump is active type of transport. Specifically, sodium and potassium ions are transported through pump in and out of the cell. Can be these ions are transported. They may be against concentration gradient. Both energy and transport proteins are required. Vascular transport is active type of transport, and liquid, solids, and larger macromolecules can be transported through vascular transport. The direction is in and out of the cell. Specifically, it is against concentration gradient. It requires energy, yes, but sometimes it doesn't require transport proteins, but sometimes it may require these transport proteins. These are summaries of the transport mechanisms through cell membrane. Thank you for watching. I need to complete chapter 3 with the following summary questions, and chapter 4 will be continued soon. Please don't forget to like, share, and subscribe the channel. And thank you once again.